So this chapter is the citric acid cycle, right? About like cellular respiration. You might also have heard of this citric acid cycle referred to as other things, right? Krebs cycle, TCA cycle, which, you know, if you notice, TCA, I don't know. Normally you don't use T as the, right? Normally leave those out, but anyways. Key topics are going to be cellular respiration, the conversion of pyruvate to activated acetate. Have you guys looked at those mechanisms, tried to draw those at all? How do you feel about this mechanism, the conversion of pyruvate to acetate? That's probably one of the toughest ones, right? Maybe we could work on that on the board too, if you guys want to try that. We're going to look at the reactions in the citric acid cycle, regulation of the citric acid cycle, and then conversion of acetate to carbohydrate precursors. Oh, I'm not even going to bother with that. I should have gotten rid of that. So, how do you think the citric acid cycle is regulated, basically? If you guys remember from last time, markers of high energetic states of the cell kind of prevent you from producing more high energy compounds. So, and then low energetic states. What's a chemical that would be a marker for a low energetic state of the cell? High concentrations of AMP. And then high energetic state of the cell, you're going to have high concentrations of ATP, right? So likewise, similar regulation as we see in glycolysis. So why do we do the TCA cycle and oxidative, or you know, oxidative respiration? Is because if we do without oxidation, we only get about 146 kilojoules per mole, and pyruvate is the result. Full oxidation, we're going to get 2,840 kilojoules per mole when we fully oxidize glucose all the way to carbon dioxide and water. So you get a lot more energy if you have oxygen available, and life is going to take advantage of that oxygen that is readily available out in the world these days. So cellular respiration is the process in which cells consume oxygen, produce carbon dioxide. It provides more energy from glucose than glycolysis, of course. Also captures energy stored in lipids and amino acids. So amino acids you can burn anaerobically just by converting them to glucose, but then you only get that partial bit of energy. But lipids you have to burn oxidatively. And to get all the energy out of amino acids, you'll need to burn oxidatively. It's got an evolutionary origin that developed about 2.5 billion years ago. It's used by animals, plants, and microorganisms. And it occurs in 3 major stages: acetyl-CoA production, where we're going to take pyruvate and make acetyl-CoA out of it, and then acetyl-CoA oxidation. And during that oxidation process, we'll accumulate electrons on electron carriers, and then those electrons will be transferred, and that's part of the electron transport chain, which will be, I think, the next chapter, so you won't see this on the test. And that results in oxidative phosphorylation. So stage 1 is acetyl-CoA production. So we generate some ATP, NADH, and FADH, right? So we take pyruvate dehydrogenase complex, we lose carbon dioxide, and that produces acetyl-CoA, which is an activated form of acetate. And then that will be able to enter the TCA cycle. Stage 2, again, generate more NADH, FADH2, and 1-GTP, which is the equivalent to an ATP. It's just a G instead of an A, right? And then stage 3 is the electron transfer and oxidative phosphorylation. We generate a lot of ATP here because we're taking all the electrons and these reduced cofactors, transporting them down. They transfer down their electrochemical gradient, and at each step's going down their electrochemical gradient. not electrochemical, reduction potential gradient. You pump protons against their electrochemical gradient. So you have a proton gradient, and as that flows down its electrochemical gradient, it causes conformational changes in an ATP synthase, which produces ATP. And we'll see that not in this chapter, but the next chapter. So in eukaryotes, citric acid cycle occurs in the mitochondria. Where does glycolysis occur? In the cytoplasm, right. So we got to get the acetate or the pyruvate into the mitochondria to perform the citric acid cycle. So citric acid cycle occurs in the mitochondrial matrix, and the oxidative phosphorylation occurs at the inner membrane. And that's where we generate this proton gradient at the inner membrane, and it generates ATP that way. So the first step is we get pyruvate from glycolysis. We have to convert that to acetyl-CoA. And the net reaction, it's oxidative decarboxylation of pyruvate. The first carbon's of glucose to be fully oxidized. So we've taken glucose and we produce carbon dioxide with one of those carbons, right? So glucose resulted in pyruvate, and then we lose one of this carboxylic acid here. And it involves all of these cofactors. This pyruvate dehydrogenase complex has 3 enzymes, but it's this huge multi-enzyme complex. They even show pictures of it, with, I think it was a scanning electromicrograph or tunneling. Where is it? Right here. These are the pyruvate dehydrogenase complexes. You can actually see them. They're huge protein complexes with many, many subunits and three enzymes that have different enzymatic functions. Okay, so coenzyme A is a cofactor, or required cofactor. So is NAD, FAD, lipoate or lipopoic acid is another cofactor, and thiamine pyrophosphate is another required cofactor for this reaction. So coenzymes, they're not a permanent part of the enzyme structure, so they associate, fulfill a function, and then dissociate. So they're not covalently bound. So that's, if you remember coenzyme A, again, you have this ADP here, really, right? So that's your handle, which is the binding site for the enzyme, where it's going to bind to the proper site. And then this is the reactive group, and we form a thiol ester with acetyl-CoA, and that's an activated form of this acetate, basically. So, and then we have this pantothenic acid. We'll see in the future, in future chapters, there's a protein that looks exactly like this, except for the handle. This is all attached to a protein, and that's involved in lipid synthesis, but that's later on. So prosthetic groups, right? They're strongly bound to the protein. Lipopoic acid is an example of this. This is the active site of lipopoic acid. It has this long carbon chain, and it has an amide bond with a lysine on the protein. So it's covalently attached to the protein, which makes it a prosthetic group, right? And there's 2 forms of lipopoic acid. You have this oxidized form, where you have basically a disulfide bond between the sulfurs. And then you have a reduced form. And if you notice, there's an extra hydrogen molecule attached to this, right? So you have a hydride and then a hydrogen ion there. And then you can have an acetylated form, and we see the acetylated form as an intermediate during this reaction, pyruvate decarboxylase reactions, or pyruvate dehydrogenase reaction. So the pyruvate dehydrogenase, it's, like I said, it's a large complex, up to 10 megadaltons in size, which is huge, right? Pyruvate dehydrogenase is the enzyme 1. Dihydrolipoyl transacetylase is enzyme 2, and then dihydrolipoyl dehydrogenase is enzyme 3. So just from the names, what do you think pyruvate dehydrogenase, what kind of activity is that going to do? Yeah, so it's an oxidative reduction reaction. So this is where you get the oxidation of pyruvate. And you also get decarboxylation at this step. What about dihydrolipoyl transacetylase? Transacetylase, what do you think that is? So in this step, it's just transferring acetate that happens to be covalently bound to the lipoic acid, from one group to another. And then dihydrolipoyl dehydrogenase, again, dehydrogenase, it's an oxidative reduction reaction. So this reoxidizes the lipoic acid, and in the process it transfers those electrons that were generated from pyruvate dehydrogenase, and it transfers them ultimately to NAD to make NADH, which will then ultimately go to the electron transport chain, right? So the advantage of the multi-enzyme complexes is you have short distances between catalytic sites. So it's what known as substrate channeling or product channeling. So it goes, you get one reaction and then it's really close in proximity, so it's just channeled to the next reaction and then the next reaction. So you don't end up getting diffusion of that product somewhere else and having side reactions or losing that product. Minimizes side reactions. Regulation of activity of one subunit affects the entire complex, right? So no reason to have the other things active if you want to shut down the complex or increase it. Like I said, these are huge complexes. You can see them with cryo-electron microscopy. So, and then this is basically the cryo-EM data, 3D reconstruction of one of the complexes. 10 nanometers. You have enzyme 1, enzyme 2, and enzyme 3, and those products get channeled through there like that. So. And this is the overall reaction, right? So you have pyruvate enters, TPP then picks up the pyruvate, and at the same time it decarboxylates that. So how does it do that? Do you guys know? Anybody? Do you have the mechanisms with you here at all? No? Do you know the structure of TPP? Probably not, right? Maybe I can pull them off of the line. Because I could draw them, but I won't be 100% correct. And we can just try to draw out this reaction. Or maybe I'll just do it this way. Actually, I can do it this way. So, do you guys remember this from chapter 14? Part of the fermentation of alcohol. So this is thiamine pyrophosphate. Okay? So we could draw this out, because this is basically the starting point of this pyruvate dehydrogenase complex. You have this thiamine pyrophosphate. Do you guys remember what's special about this here? That hydrogen?
Is that the one where the hydrogen leaves off and then the ring opens?
No, the ring doesn't open. The hydrogen, I mean, you have a nice positive charge here, which kind of allows for this to be an acidic hydrogen. So that hydrogen, just the proton itself can dissociate really easily. And this quaternary nitrogen stabilizes negative charge here on a carbocation. So you get dissociation. Wrong button. Let's see. So you stabilize this negative charge on the thiamine pyrophosphate, which, now that you have this carbanion, you can perform nucleophilic attack on that carbonyl on the pyruvate, which is what happens. This is the first step of this pyruvate dehydrogenase. And then you have this pyruvyl thiamine pyrophosphate, okay? So this here acts as a great electron sink. It's a quaternary nitrogen again. You guys remember there's other things that are great electron sinks with the quaternary nitrogen. We see that with the, what's it called? Think of the NAD, right? It's a great hydride acceptor because of that quaternary nitrogen. So anyways, now we have pyruvyl TPP. These electrons from the carboxylic acid will drop down, and that makes what? Carbon dioxide right here. The other electrons have to go somewhere, and this quaternary nitrogen is a great electron sink, and you end up with a structure like this, okay? So TPP then stabilizes this carbanion resin, you get this resonance stabilization. But now here is where we get to pick up the, I gotta find the right marker, right? So let me draw this. I'm a terrible artist. So
Just go away.
exactly. Oh, that one's not very good either. So, oh, that one's not bad. So we have the R, this quaternary nitrogen, or we'll leave it non-quaternary now, but we'll show the things here. Sulfur here. And then we have to so I'll stop it here, go back to the other PowerPoint. Doesn't want me to do anything. PowerPoint failed. So we've basically oxidized what here? The pyruvate. We let carbon dioxide has come off. Okay, but what's going to happen now is you have lipoproic acid. I can't draw, can I? And there's, it's on a long chain like that. Now it's got electrons here, right? So what can happen next is, so like, hold on, where? It just opened me up a blank one. So, like in that resonance structure, these electrons drop down, these electrons can come out. They can attack this, leaving these electrons to come pick up a proton. Okay, and then you end up with this structure with TPP. It's got a SH, and then an S, and then, oh, I'm missing the methyl group here. See, I just flipped that around. So just to show that this lipoproic acid is now bound to what was the pyruvate, right? Okay, another base can pick off this hydrogen. These electrons can drop down, and then these electrons can stay with the TPP. And then you end up with lipoproic acid bound to plus the free TPP okay. So now we have this, right? And then the next enzyme is the what? No, not that one. The dihydrolipoyl transacetylase. So we have acetyl-CoA, so CoA-SH. These electrons can come and attack this carbonyl, leaving these electrons to pick up a hydrogen ion. And then you end up with acetyl-CoA. And that will leave as acetyl-CoA. And then you have lipoic acid. That's reduced, right? The last enzyme, enzyme 3, the portion of this complex, dihydrolipoyl dehydrogenase, and what that does is it regenerates the oxidized form of lipoproic acid. And in reality, it transfers these electrons to FAD as part of the complex, and then eventually it ends up at NAD. So, boy, I don't know if I can draw FAD. Should I try? Or what we can do is like this. So you have F. These electrons go to here, and then, you know, this hydrogen. So these electrons attack here, these electrons go to FAD, and you end up with FAD. H2, and then the regenerated lipoic acid, oxidized form. And then this FAD will transfer its reaction to NAD to make NADH, which is unusual. Normally, the electrons will never go from FADH2 to NADH, but the way it is in this enzyme complex, it is going down its reduction potential just because of the special way that FAD is part of this complex. So this is, I don't know if you can understand my artwork here. It's pretty bad, but basically this is the mechanism, right? I didn't draw out the decarboxylation, but it's pretty clear in the animation, I think. What do you think? The animation does a better job at drawing than I do. So, but this reaction is basically the same thing that we're going to see in alpha-ketoglutarate decarboxylase, or not, dehydrogenase. Alpha-ketoglutarate dehydrogenase is part of the TCA cycle. So now this acetyl-CoA is then free to enter the TCA cycle, right? So, and you can see here, TPP, this hydroxyethyl TPP, lipoic acid picks it up. Then you have acetylipolysine. Coenzyme A picks up the acetate group from the lipoic acid. You have reduced lipoic acid, then has to be reoxidized. That's reoxidized with FAD to produce FADH2, and then NAD will ultimately pick up those electrons from the FADH2. So, is that okay? So sequence of events in oxidative decarboxylation, we just went through all of these. First, decarboxylation of pyruvate to the aldehyde, the acetylaldehyde. Next is oxidation of the aldehyde to a carboxylic acid, and those electrons those electrons reduce lipomide and form the thioester. Now you have a thioester, it can be transferred to acetyl-CoA, and then you have to reoxidize the lipoic acid. And in the process, you generate an FAD cofactor, which will ultimately transfer those electrons to NADH. Same thing happens in alpha-ketoglutarate dehydrogenase. And then this acetyl-CoA can then enter the TCA cycle or the citric acid cycle, or here it's the CAC cycle, or TCA. So many different names. So acetyl-CoA, first step is citrate synthase. So that's an enzyme, tells you it synthesizes citrate from acetyl-CoA and oxaloacetate. Then we go to aconitase, which just rearranges the alcohol group to form isocitrate. And then we get isocitrate dehydrogenase, where we generate an NAD, and it's a decarboxylation that forms alpha-ketoglutarate dehydrogenase complex, which is exactly like the pyruvate dehydrogenase complex, just uses alpha-ketoglutarate as a substrate rather than pyruvate, which will form succinyl-CoA synthetase, which this is one of those enzyme names where it's kind of named for the reverse reaction in what we actually see. So you take succinyl-CoA and we produce a GTP and succinate. And then succinate dehydrogenase forms fumarate. Fumarase just, you can't see, but there's a double bond there. Just hydrates that double bond to form an alcohol, to form malate, and then malate dehydrogenase, again we generate another NADH, and that regenerates oxaloacetate, which can enter, bind with or react with acetyl-CoA again via citrate synthase to produce citrate again. So sequence of events in the citric acid cycle. First, we're going to form a carbon-carbon bond to make citrate between the acetate and the oxaloacetate. Then we're going to isomerize via dehydration-rehydration reaction, and just moving that alcohol group from a tertiary carbon to a secondary carbon. And that makes it good for a decarboxylation, an oxidative decarboxylation on the next step. Step 5 then we have another oxidative decarboxylation, alpha-ketoglutarate, which looks very much like this, just different substrates. Substrate level phosphorylation with the succinyl-CoA, instead of generating acetyl-CoA, you generate succinyl-CoA, an activated succinate. And the energy from that CoA group can generate GTP. Step 6 is dehydrogenation to reduce FADH. And then this step, this oxidative reduction reaction is actually part of the electron transport chain. It's directly into the inner membrane of the mitochondrial matrix, and these electrons go directly into the electron transport chain. Then we get hydration of the double bond, and then dehydrogenation to give NADH and regenerate oxaloacetate. So the first step is citrate synthase. We take acetyl-CoA, react it with oxaloacetate, and we generate citrate. So, condensation of acetyl-CoA and oxaloacetate, the only carbon-carbon bond formation we get here. Uses acid-base catalysis. Methyl of acetyl-CoA is not a good nucleophile unless it's activated by deprotonation. So you got to deprotonate something here, meaning acid-base catalysis. The rate-limiting step for the citric acid cycle. Activities largely depends on oxaloacetate. It's highly thermodynamically favorable, so it's irreversible. So this is going to be one of the regulated steps, right? Regulated by substrate availability and product inhibition. And if you remember, a lot of the steps in glycolysis are regulated by the same product, right? Citrate concentrations, when we see citrate concentrations build up, that's going to inhibit entry into the glycolysis cycle. No, that's not a cycle, just glycolysis. So induced fit in citrate synthase. So this is an example where we have clear different conformations, right? So, and what it does is it avoids unnecessary hydrolysis of that thioester acetyl-CoA. That's an activated, that's a high energy thioester. You don't want to lyse that acetyl-CoA if you don't need to. So, and you wouldn't want to do that if you don't have oxaloacetate bound. So in the open conformation, the free enzyme does not have a binding site for acetyl-CoA. It has to bind to oxaloacetate first, and then that causes a conformational change, opening that binding site for acetyl-CoA. So you're not going to burn that, thioester in the process without oxaloacetate ready there for that carbon-carbon bond. And this shows you crystal structures with the open conformation and the closed conformation. So, and then the oxaloacetate can bind in. It causes the closed conformation, allowing a binding site for acetyl-CoA. And this next step is the mechanism for citrate synthase. So here we got two histidines and an aspartic acid. The aspartic acid acts as a general base, picking off the hydrogen from the methyl group. And you end up with an enol intermediate. The electrons from that carbonyl group on the carboxylic acid come up and pick up the hydrogen from a histidine, and you have an enol intermediate. And then what happens next? The histidine picks back off, gets that proton back. These electrons drop down, leaving these electrons have to go somewhere, and they perform nucleophilic attack on the carbonyl on the oxaloacetate. and you have a citryl-CoA intermediate. So you still have this CoA bound, and it's a citryl-CoA intermediate now. Water will then come in and hydrolyze that thioester bond. Releasing coenzyme A. Eventually when it drops back down. So you release coenzyme A, and then you have citrate as the product. And then you've got the mechanism here on the PowerPoint. And I think it's in your book too, right? So. Next reaction is to isomerize. You take citrate and make isocitrate. Basically, you're just moving the alcohol from this carbon to this carbon. It makes it next to this carboxylic acid. Okay, that makes it easier for a decarboxylation, reduction or oxidation to occur. So, elimination of water from citrate gives a cis-ene bond, which is a lyase reaction. Citrate is a tertiary alcohol's poor substrate for oxidation. Isocitrate, being a secondary alcohol, is a good substrate for oxidation. You have that oxygen there. Additional water to cis-aconitate is stereospecific. Thermodynamically unfavorable, it's reversible. The product concentrations are kept low to pull it forward. So it's going to run in reverse. It could run in reverse if the product concentrations build up, and it would. So, in water removal substrate, the addition to cis-aconitate are catalyzed by this iron sulfur center, basically. And it's stereospecific. Only R isocitrate is produced by aconitase, distinguished by the 3-point attachment to the active site. So you recognize enzymes are often highly stereospecific. Next reaction is oxidative decarboxylation number 2 from isocitrate dehydrogenase. You have isocitrate. It's oxidized by the hydride transfer to NAD or NADP, and the isocitrate dehydrogenase isozyme. So you have isocitrate, basically, and I think we got the mechanism here. Yes. So we lose carbon dioxide, generate NADH. Oxidation of the alcohol to a ketone transfers a hydride to NAD. Cytosolic isozyme uses NADP as a cofactor, and that generates a reducing equivalent for anabolic reactions. Highly thermodynamically favorable, irreversible, regulated by product inhibition. And go through this mechanism. So you have isocitrate. NAD is going to grab this hydride, which will form a carbonyl. The alcohol drops down. Manganese stabilizes this. These electrons drop down, you lose carbon dioxide, you form this ene intermediate. And you have an oxide there in its place. This oxide, the electrons drop down to form the carbonyl again, or the ketone, and these electrons have to pick up that hydrogen ion. And then you have alpha ketoglutarate. So you have alpha keto, this is keto group on the alpha carbon, and a carboxylic acid, and it's glutarate. This ends up being the precursor for glutamic acid. You add an amine group there, and you get glutamic acid. So the final oxidation decarboxylation is alpha ketoglutarate dehydrogenase complex, which is basically the same mechanism as the pyruvate dehydrogenase complex. So you have alpha ketoglutarate, coenzyme A and NADH come in. Cofactors include thiamine pyrophosphate, and it's basically the same mechanism. It's an odd picture of alpha ketoglutarate that the book gives you there, right? That doesn't look quite right, does it? First of all, this should be the carbonyl here, and you should have a single carbon-carbon bond there. So, sorry about your book's mistakes. Huh?
Yeah, that's what that was.
Yeah, that. So you have a double bond here. You got two oxygens. Actually, one of these is going to have a double bond on the carboxylic. That'd be 5 bonds for this carbon. Does that make sense?
Yeah.
And then this should have a charge the way it's written here. So that's a bad figure. So alpha ketoglutarate dehydrogenase, it's the last oxidative decarboxylation. You net full oxidation of all carbons of glucose after 2 turns of the cycle. So carbon is not directly from glucose because the carbons lost came from oxaloacetate, but you get 2 turns of the carbon of the cycle, and you'll lose those carbons that entered 2 cycles beforehand. Succinyl-CoA is another high-energy thioester bond. It's highly thermodynamically favorable, irreversible, regulated again by product inhibition. And this shows you the origins of the carbon atoms in CO2. So you have citrate, right? So this is the acetyl-CoA that's added on the end. Isocitrate are still there. And remember, we lose this carbon dioxide here. And then alpha-ketoglutarate is here. And again, we're losing this carboxylic acid. So we haven't, these are the original carbons that have come in from acetate. So we haven't lost those yet. Okay? You lose those later on. And again, like I said, the alpha ketoglutarate dehydrogenase, same coenzymes and identical mechanisms to the pyruvate dehydrogenase. So you have alpha ketoglutarate, produces succinyl-CoA, same exact mechanisms. You have lipoic acid and thiamine pyrophosphate. That carbanion attacks the keto group, causes decarboxylation of this, and then lipoic acid picks it off of the thiamine pyrophosphate, and then that transfers it to coenzyme A, forming succinyl-CoA. And then the NAD gets the electrons regenerating the lipoic acid. So the next succinyl-CoA synthase generates GTP, a high-energy phosphate compound, from the thioester bond, the succinyl-CoA, and generates succinate. So succinyl-CoA, and this is substrate level phosphorylation, which is different than oxidative phosphorylation that we get at the inner mitochondrial membrane. Substrate level, do you guys remember there were two reactions where you have substrate level phosphorylation in glycolysis, right? Pyruvate kinase, if you remember, takes phosphoenolpyruvate. That phosphate from phosphoenolpyruvate gets transferred to ADP to make ATP, substrate level phosphorylation. Also, what's it called there? You take, do you remember the other reaction off the top of your head? So we have GAPDH, right? So glyceraldehyde 3-phosphate dehydrogenase. And then the step after that, I can't remember right now. Don't worry about that. But remember what substrate level phosphorylation is where that phosphate gets transferred directly to GTP rather than it oxidatively being phosphorylated based on a proton gradient at the inner mitochondrial membrane. So the energy of the thiol ester allows for this incorporation of inorganic phosphate. It's a high energy, so phosphate will come in and replace that. Goes through all the phosphoenzyme intermediate, produces GTP, which then can be converted to ATP. It's slightly thermodynamically favorable, reversible. The product concentrations are always kept low. So what happens, there's a histidine on the enzyme, and succinyl-CoA will come in. The histidine on the enzyme picks up the phosphate. The phosphate then transfers to the succinyl, so you have an enzyme-bound succinyl phosphate then. That succinyl phosphate can then transfer that phosphate to the histidine on the enzyme. You have a phosphohistidine enzyme, then that transfers the phosphate to GDP from the histidine. So phosphate comes in and replaces the coenzyme A. The phosphate then gets transferred to the histidine at the active site, releasing succinate. Phosphohistidine then transfers the phosphate to the GDP to make GTP, and you get GTP released and regeneration of that enzyme. Whoa. So, next reaction is succinate dehydrogenase, where we take FAD, reduce it to FADH, and this is part of the electron transport chain. Remove one of these hydrides, these electrons will drop down and you form a fumarate, or you have an ene group there. So like I said, this is bound to the inner mitochondrial membrane. It's part of complex 2 in the electron transport chain, reduction of alkane to alkene. It requires FADH as the hydride acceptor. Reduction potential of NAD is too low, so FAD is covalently bound. Near equilibrium, reversible. The product concentrations are kept low to pull it forward again. So this isn't a highly regulated step. You're near equilibrium, if you remember from last time. So the next reaction is fumarase, where we add a hydride across this, not a hydride, hydroxide across the double bond. You have a carbanion transition state. Hydrogen ion comes in and to pick up that hydrogen ion from the carbanion, and you end up with L-malate, where we've added this alcohol across the double bond. And then fumarase, which is what we just did. We add the water. Always transforms L-malate, hydroxide adds to fumarate, then a hydrogen ion adds to the carbanion. Cannot distinguish between inner carbons, so either can gain the hydroxide. Slightly thermodynamically favorable, reversible. Product concentrations are kept low again, pulls that reaction forward. And this shows you the stereospecificity of fumarase. So you have fumarate, can produce L-malate or D-malate. So then we have the oxidation of the alcohol to a ketone by malate dehydrogenase. NAD picks up this hydride. The electrons from this oxygen will drop down. To fill that in, you'll reform oxaloacetate then. And then that ends the cycle, basically. You generate NADH. It's the final cycle. Final step of the cycle regenerates oxaloacetate. It's highly thermodynamically unfavorable. It's reversible. Oxaloacetate concentrations are kept really low, though, and that's what pulls this reaction forward, making it favorable, because the super low concentrations of oxaloacetate. So in one turn of the citric acid cycle, acetyl-CoA becomes citrate, or forms with oxaloacetate, becomes citrate. Transition that alcohol from the tertiary carbon to the secondary carbon, so to form isocitrate. Then you get oxidative decarboxylation, forming NADH, carbon dioxide, and alpha-ketoglutarate. Alpha-ketoglutarate gets oxidative decarboxylated again with this mechanism, forming succinyl-CoA, which can then perform substrate-level phosphorylation. Inorganic phosphate substitutes the coenzyme A, which then gets transferred to the histidine on the enzyme, which can transfer it to GDP to make GTP. Succinate then becomes oxidized to form the ene of fumarate, and you form FADH2, and those electrons go directly into the electron transport chain. Fumarate gets hydrated to form malate. Malate gets oxidized. The alcohol on the malate gets oxidized to form oxaloacetate. And then that cycle gets repeated over and over again. Net results, 1 acetyl-CoA plus 3 NAD, FAD, GDP, phosphate, 2 waters. You get 2 carbon dioxides, 3 NADHs, and FADH2, GTP, coenzyme A, and 3 hydrogen ions. Next oxidation of 2 carbons to carbon dioxide, equivalent to 2 carbons of acetyl-CoA, but not the exact carbons that entered the cycle. They had entered the cycle previously. Energy captured by the electron transfer to NADH and FADH2. We generate 1 GTP, which can be converted to ATP, because they're basically equal in energy. And then you get completion of that cycle. Now, I'm not going to quiz you on numbers of ATP produced of these overall cycles, like they do here. So the indirect and direct ATP yield. So ultimately the number of ATP produced, we always see this as 30 is 32. I think they probably make you memorize that in core bio or microbio, but I'm not going to make you memorize the ultimate number. Counting ATP. I like the mechanisms better, which is more fun, right? So citric acid cycle intermediates are amphibolic. So what does that mean? They serve multiple different roles in metabolism, right? So citrate can form fatty acids and sterols. Alpha-ketoglutarate, like I mentioned previously, is the precursor to glutamate or glutamic acid, which is the precursor to your purine bases and the precursor to arginine, proline, and glutamine. So you can make a lot of amino acids from alpha-ketoglutarate. Succinyl-CoA is a precursor for porphyrins. Do you guys remember what porphyrins are? The porphyrin rings are what make up heme and hemoglobin and myoglobin. Malate you can make pyruvate can be formed into malate via maleic enzyme. Oxaloacetate is the precursor to aspartic acid, asparagine, and the pyrimidines. Okay. Oxaloacetate can also form phosphoenolpyruvate, right? If you guys remember the first step for gluconeogenesis, we what? Pyruvate. We carboxylate pyruvate, right? To form oxaloacetate. That's why the arrow is here, going pyruvate to oxaloacetate. You add a carboxyl group to form oxaloacetate, and then that oxaloacetate is decarboxylated and phosphorylated, and you form phosphoenolpyruvate, right? Do you remember from gluconeogenesis? And then that phosphoenolpyruvate can form serine, glycine, cysteine, phenylalanine, tyrosine, and tryptophan, precursor for nearly all the amino acids. Or it can be used to form glucose and used as an energy source, stored as glycogen. So that's what they mean by amphibolic. There's a lot of the intermediates in the citric acid cycle, they're used for a number of purposes in metabolism, precursors. And then they have these aneuploitic reactions, which are like the filling reactions. So when you run low on the TCA intermediates, these aneuploitic reactions can refill those intermediates. The one we've seen before is the pyruvate carboxylase, right? That's the one of the first steps in gluconeogenesis. But if you're low on oxaloacetate, you need that to burn acetic acid to generate ATP. You can form pyruvate, you can form oxaloacetate from pyruvate via pyruvate carboxylase. Tissues that do this are the liver and the kidney. Phosphoenolpyruvate plus carbon dioxide and GTP, PEP carboxykinase forms oxaloacetate in the heart and the skeletal muscle. Phosphoenolpyruvate and bicarb form oxaloacetate here. Higher plants, yeast, PEP carboxylase. Again, these are all enzymes that generate oxaloacetate. And then, of course, oxaloacetate can condense with acetic acid, form alpha-ketoglutarate down the way, which can form glutamic acid and so forth. Pyruvate, here, malic enzyme, you can form malate from pyruvate, and that's widely distributed in eukaryotes and bacteria. So, and these reactions are there to replenish the intermediates if for some reason they get depleted. And as we saw with pyruvate carboxylase, remember? Biotin is the important cofactor in this, right? So biotin is a carbon dioxide carrier. And we get the carbon dioxide for this reaction from bicarbonate. So bicarbonate, before it can be added, has to be activated. So it's activated via phosphorylation by ATP. So you get activation of bicarbonate to form carboxyphosphate intermediate, and then that carboxyphosphate can transfer that carboxyl group directly to the biotin. And biotin's, do you guys remember how I described it? It's like on a tether, and it can swing from one subunit of an enzyme to another subunit to transfer that carboxy group. So now you have the carboxylic acid, and the CO2 is on the biotin, and you have carboxybiotinyl enzyme. The biotin transports the carbon dioxide from one active site to the other. Biotin, and then it can be added to pyruvate here to produce oxaloacetate. And I think we gotta look at this. Wait, this isn't the right one, is it? We already saw this one. It must be messed up. Yeah. doesn't work at all. Anyways, I just talked you through that, right? Oh no. PowerPoint doesn't like me. There we go. So you see there's two different catalytic sites, 1 and 2, and then a biotin covalently bound to a lysine here. Bicarbonate has to be activated to carboxyphosphate via ATP, phosphorylation via ATP. And then the next thing that will happen is you'll get transfer of that carboxy group to the biotin. So then you have carboxylated biotin, and it'll swing from catalytic site number 1 to catalytic site number 2, where it'll transfer that carboxyl group to this next substrate, which will be pyruvate. See there, it's swinging, or in this case, it looks like it rotated. And then pyruvate will pick up the carboxylic acid forming oxaloacetate. And you have oxaloacetate as the product with the bicarbonate carboxylic acid added to the pyruvate. So, and that's the thing, if we notice biotin and lipoic acid both are tethered to proteins via long carbon chains of lysine, right? And then there's a further carbon chain to the active part of those coenzymes. We also see a nice tether sort of from on this acyl carrier protein. Remember how before the beginning of the lecture I told you about acetyl-CoA? This is the active part of the acetyl-CoA. And in fact, lipid synthesis, we have an acyl carrier protein rather than acetyl-CoA because it's all part of, tethered to that protein, the production of lipids. But the purpose of these tethers is that it allows flexibility for that enzyme to transfer groups from one place to another to get further reactions on. So just remember that certain ones are on a tether. So. Regulation of the citric acid cycle, again, if your cell is a high energetic state, you don't want to oxidize energy to produce more energy. You got plenty of energy. So high NADH, succinyl-CoA, citrate, and ATP are all going to inhibit acetyl-CoA entering the TCA cycle via citrate synthase. So this is the heavily regulated step. ADP actually activates it. Normally you see AMP, but ADP will activate citrate synthase because that's an indication of the low energetic state of the cell. And you remember pyruvate dehydrogenase is inhibited again. ATP, acetyl-CoA, NADH, and fatty acids are all going to inhibit pyruvate dehydrogenase. So again, high energetic state of the cell, you don't want to send things into the TCA cycle. Low energetic state of the cell, you're going to activate and send things into to be burned to produce more energy. So then isocitrate dehydrogenase, ATP inhibits it again. Calcium and ADP activate it. Alpha-ketoglutarate dehydrogenase, succinyl-CoA, product inhibition. NADH and inhibit it, and calcium activates it. And then we have, and those are the regulated steps right there. Everything else, if you remember, is kept really close to equilibrium, so it runs in reverse or forward, and it's all based on product concentrations, which direction it's going to flow. So, and like I said, it's regulated the highly thermodynamically favorable and irreversible steps. Pyruvate dehydrogenase, citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. And general regulatory mechanisms activated by substrate availability, inhibited by product accumulation. Overall products of the pathway are NADH and ATP. Right? So affect all regulated enzymes in the cycle, inhibitors, NADH, and ATP, activators, NAD, and AMP. So the regulation of pyruvate dehydrogenase is also regulated by reversible phosphorylation of enzyme 1. Phosphorylation, if you have phosphorylation, it's inactive, so it's not going to work. Dephosphorylation, it's active. So what phosphorylates enzyme 1 is pyruvate dehydrogenase kinase, right? Simple name. If you remember, kinases add phosphate groups. This kinase adds phosphate group to pyruvate dehydrogenase. And pyruvate dehydrogenase phosphorylase are part of the mammalian pyruvate dehydrogenase complex. The kinase is activated by ATP, kinase activities. So if you have high ATP concentration, it'll activate this kinase activity, phosphorylating enzyme 1, inhibiting, enzyme 1, so making it inactive. Low ATP, the kinase is less active, and phosphorylase removes the phosphate from the PDH, and you get more acetyl-CoA generated. Additional regulatory mechanisms, citrate synthase is also inhibited by succinyl-CoA. Alpha-ketoglutarate is an important branch point for amino acid metabolism. Right? So succinyl-CoA communicates the flow at this branch point to start the cycle. So if you have a lot of citriol-CoA generated, you don't need to send more, oxaloacetate to be made into citrate. Regulation of isocitrate dehydrogenase controls citrate levels. Aconitase is reversible, if you remember. Inhibition of isocitrate dehydrogenase leads to the accumulation of isocitrate, and that reverses aconitase, and you'll accumulate citrate then. And that leaves the mitochondria and inhibits phosphofructokinase 1 in glycolysis. If you remember, that's the committed step in glycolysis. And if you remember, it's regulated by high citrate. So if the TCA cycle gets backed up, aconitase runs in reverse. You take isocitrate and make citrate. Citrate leaves the mitochondria and inhibits phosphofructokinase 1, which is the entry step into glycolysis. And that's basically it for the TCA.